Hello, welcome to this channel, and today we'll be discussing about a very important part from plant physiology, and that is floral development. Now, the genes which are associated with the floral development are homeotic genes and MAD-S box genes. These two things we should remember. In these homeotic genes, there are basically three different type of genes. First is floral timing gene, which determine the timing of the flower development. We also call them FT genes, and the locus where the FT genes are present are known as FT locus. The second one is floral identity genes. Now these genes, they are epitala 1 or AP1. Second one is epitala 2 or AP2. Third one is leafy, and the fourth one is cauliflower. We also call this cauliflower as cal. What they do, they develop this shoot apical meristem into flower. So they convert the shoot apical meristem into flower. The third type of gene is organ identity genes. Now there are 5 different types, and they are associated with the development of different floral organs like sepal, petal, stamen, and carpel. And in flowers, there are 4 different holes, like 4 different circles are present during the development of the flower. So this is second, this is the third, and this is the fourth. And each circle we call them as hole. So this is hole 1, and that will be developed into sepal. This is hole 2, that will be developed into petal. The third one is hole 3, that will be developed into stamen. And the fourth one is the hole 4, that will be developed into carpel. And this development, these different organs will be developed by organ identity genes, and there are 5 different types of organ identity genes, and they are epitala 1 or AP1, AP2, AP3, pistillata or PI, PT, and agamus or AG. So epitala 1, epitala 2, these two are the common members of both floral identity genes and organ identity genes. So if we look further in organ identity genes, there are 5 different genes: epitala 1 or AP1, epitala 2 or AP2. Then the third one is epitala 3 or AP3. The fourth one is pistillata or PT or PI in some books, and agamus or AG. Now interesting thing is, this epitala 1 and epitala 2, they are being encoded by the class 1 genes, where epitala 3 and pistillata, they are coded by class B genes, and agamus is coded by class C. This is not 3, this is class C genes. Okay, so pistillata and epitala 3, they are coded by class B, and epitala 1, epitala 2, they are being coded by class A genes. And if we have already studied about the 4 different holes, so if class A is present, epitala 1 and epitala 2 genes will be there, and they will be developed into sepal or hole 1. Let's just copy this figure and paste it. Right, so class A is associated with the formation of sepal or hole 1. When both class A and class B is present, so there will be AP1, AP2 from class A, and AP3 and pistillata from class B. Then petal will be developed, and that is the hole 2. So this is the hole 2. The next is when both class B and class C genes are present. So there will be both AP3, PT, and agamus, and they will be developed into stamen or hole 3. So this is hole 3. And when only class 3 genes are present, that means only agamus is present, that will be developed into carpel or hole 4. So that is hole 4, associated with only class C. Now these things are very important while studying the deletion mutation. Okay, now let's say we have this is class A, C and B genes. When class A is only present, that will be developed into sepal. When A and B both are present, that will be developed into petal. When B and C both are present, that will be developed into stamen. And when C only is present, that will be developed into carpel. Now here one thing is very important, that class A and class C, these are antagonistic. That means when class A is present, class C will be absent. If class C is present, that means class A is absent. And this thing is very important in studying the deletion mutation experiment results. Let's say first case class A is deleted, and we are taking again hole 1, hole 2, hole 3 and hole 4. So in the first case, C will be there because class A is deleted, that means class C is present, so C will be there. In the second phase, instead of A plus B, there will be B plus C and rest will be same. So hole 1 it will be carpel, 2 and 3 that will be stamen, and hole 4 that will be again carpel. Then class B deletion, so again hole 1, hole 2, hole 4. Okay, hole 1 A is present, so that will be developed into sepal. In the second, again sepal is developed. For 3 and 4, since B is absent, so both carpel will be developed. And in the class C deletion, in hole 1 A will be present obviously, so that will be developed into sepal. In B, in hole 2 petal, and in hole 3 again A plus B, because C is negative means A is present, so that is a petal. And in hole 4 again C is negative means only A is present, so sepal. So that is the thing about ABC model. Hope you liked it, and please don't forget to like, share, comment and subscribe. Okay, thank you.